Hello everyone, welcome back to my channel. Now in this video we are going to discuss and talk about the Griffiths experiment. So this is basically the experiment when the search for the genetic material was going on. Basically, when Mendel did the experiment for the first time and he discovered that there are some factors who are carrying some characteristics which can be inherited from one generation to the next generation. Right after that, among the scientists, it was the biggest news that they have to search for the genetic material, that what is that factor which is carrying all the information that can be inherited from one generation to the next generation. So in the year of 1928, as I have written here, you can see that in the year of 1928, Frederick Griffith was the scientist who performed one experiment, from whose experiment about the horizontal gene transfer among the microorganisms got cleared. Now, what is the meaning of all of this? Let's begin with this video. So, there are basically transfer of the gene or transfer of the factor which can carry all the genetic informations from one generation to the next generation. That can happen normally. So, from parent, say for example, from parent to the child, that means from one generation to the next generation. In this way, the characteristic can be inherited. Okay. From one generation to the next generation, all the characters can be inherited. This kind of transfer of the gene or of the factor which is carrying all the information, that is considered as vertical transfer of the gene. So, this type of gene transfer is known as vertical transfer. This was so natural and this was known by all that this is very common in each and every organism. We can find the vertical transfer of the gene or of the factor which is carrying all the genetic information. But from Frederick Griffith's experiment, another type of gene transfer or DNA transfer mechanism was cleared. The idea of that type of transfer was cleared. What is that type of transfer? Let us see that this is also possible. Say for example, this is parent 1, from where in the child, in its child, the genetic informations are being carried or transferred. And say for example, this is another parent, parent 2, and from here to its child, once again through the vertical transfer method, the characteristic features are going from one generation to the next generation. But this is also possible if these two are belonging from the same species, from the same characteristic feature and that kind of organism, then it is possible that they can transfer their characters among themselves. That means from this species, the character can go into this individual, and from this individual the characteristic feature, that is the gene which is carrying all the characteristic feature, that can be transferred from this parent to this parent or from this child to this child. That means if within the individual the transformation or the transfer of the genes are possible, and that type of transfer is known as horizontal transfer. So this horizontal gene transfer or horizontal gene transfer among the microorganisms, basically in the bacteria we can see this kind of transfer, this horizontal gene transfer. This was first being observed by Frederick Griffith's experiment. Okay, now let's see that what was the experiment of Frederick Griffith. And basically he was searching for the genetic material, but he was searching for the genetic material, but he was unable to talk about the genetical concept behind his experiment, behind the result of his experiment. Now let us see with the what was the Griffiths experiment. Okay, so basically Griffith did his experiment with one bacteria. The name of that bacteria was Streptococcus pneumoniae, which can cause the disease pneumonia. Now, say for example, there are basically two strains which are available for the bacteria. Now, first let me write down here the name Streptococcus pneumonia was the bacteria with which Frederick Griffith did his experiment. Now, in this bacteria, there were two strains which were available. In case of one bacteria, say for example, this is the bacterial body and there is no extra covering from the outside. That means there is lack of capsule. That type of strain will be very rough. When they will be colonized, they will be plated. Say for example, if this is the plating where their exact nutritional medium, temperature, pH, everything will be provided for their growth, then we can find that they will provide their rough colonies. And that is why this strain is also considered as rough strain, as they do not have any kind of smooth capsule outside them. And another type of strain was also there, where we could find that from the outside there was one capsule which was available. And as there was one capsule from outside, that is why this particular strain was forming very smooth colonies on that plate, on that nutritional medium. And as they were forming smooth colonies, that is why the strain name was given like S strain or the smooth strain. Now, there were two basic characteristic feature difference between these two strains. What are the differences between them? The R strain, as there is no extra capsule, no extra layer outside them. So what was happening to them when they will infect some host organism, say for example, they will infect into the human body, they can not cause any kind of disease because our lymphocytes, or we can say our WBCs, can very easily recognize them due to not having any kind of outside extra membrane upon them. So that is why they will be killed very easily by the defense cells of our body. And the same mechanism can happen within the rat cells also, within the rat's body also. And with which thing this capsule is made up of? Okay, I forgot to tell this. With carbohydrate, or we can say polysaccharides, the capsule is made up of. So this capsule is basically made up of polysaccharide. Okay. So as there is the polysaccharide capsule outside the body of the S strain, so that means we can take one good example that this person, say for example, two terrorists are there. One terrorist who is just moving around without having any kind of cover on his face, and another one terrorist is there, or another one criminal is there who is covering his face. So what will happen? Police will recognize this one first, and this one they will not be able to recognize. So similar kind of thing happen when these bacteria will infect the host organism or the rat cell or the human cell. Then what will happen? The defense cell will kill them. That is why this R strain is very much a virulent strain. This R strain was also considered as a virulent strain because they were unable to cause any kind of disease inside the host body. On the other hand, in case of A strain, this strain is known as virulent strain. Why so? Because this can cause disease, and this can not only cause disease, this can not only cause pneumonia, this can actually kill the organism, this can actually kill the rat. Okay, so that is why this is also known as our S strain, because or the virulent strain, because this is actually hiding its total body under some cover. What is the name of that cover? That is the capsule, that is made up of polysaccharide. So as it is hiding its face, that's why the police, that is the defense mechanism of the host body, that is completely unable to recognize them. And if they will not be able to recognize, then there will be no chance of killing. So in that way, this A strain or the virulent strain is absolutely capable of making the disease as well as kill the host organism. So this is the basic fundamental characteristic of the Streptococcus pneumoniae and of these two strains. Now what Frederick Griffith did with these two strains, he just grew these two strains on the culture plate. He separated them out and he took rat as the medium for doing this experiment. So in the first case, what he did, just see that with the rat, he just injected the R strain of the Streptococcus pneumoniae. R strain. R strain means avirulent. Avirulent means it is not at all harmful for the body. So what it will lead to? It will lead to the smiling face. That means the rat was completely alive. Rat was alive. That is why it is very happy. In the second case, what he did, it was expected because it's not having any kind of capsule. So when he injected this R strain into the rat's body defense mechanism can very easily recognize this R strain, and that can kill very easily the R strain bacteria, and that is why there will be no chances of any disease, and in that way the rat will be alive. In the second case, what he did, he took another rat for his experiment. He injected the S strain. So in this case, when he injected the S strain, now S strain is hiding his face under the cover, that is under the capsule. So that is why what will happen, the rat will die eventually because due to having this disease. So death of the organism was occurring. Death of the rat was occurring. In the third case, what Griffith did, he took another rat and he injected the heat-killed S strain. Heat-killed S strain means, what is the meaning of heat-killed S strain? Let's suppose here some heating experiment was given to this bacteria. So what will happen? It will just kill all the outside everything. So what will happen, once the capsule will not be there, when the mask will not be there on the terrorist or on the criminal, then police will very easily recognize him, and police will very easily arrest and kill him. So similar kind of thing was happening in this case. When the heat will be provided to the S strain, at the first step, what will happen? The capsule will disappear. As there will be no presence of capsule, so it will be very easy for the rat's body defense mechanism to recognize it and to kill it. So in that way, the S strain will be killed. And in this case also, the rat will remain alive. There will be no chance of any disease and rat will remain alive very happily. But in the fourth case, what will happen? There comes the twist of this experiment. That is, with the rat, he provided the R strain along with the heat-killed S strain. Along with the heat-killed S strain. So what it was leading to? Now what he is doing? He has provided here the R strain. That is, say for example, this is the R strain now, and this is the S strain, outside which there is the presence of the capsule. Now what was happening, as this was heat killed, so there was no such this material present there. But he saw, surprisingly, the result was very surprising, that the rat was died. Rat was died. But why this death of the organism was happening? Why this death of the organism was happening? It must not be there, because R strain cannot cause any disease because it doesn't have any kind of capsule upon it. Then S strain, it can cause disease, but as he was providing heat-killed S strain bacteria, that means it would not be able to produce any kind of disease, as well as it would not be able to kill the rat. But then also, after mixing both of them, then also the death of the organism was happening. That means there is something which is carrying the information of forming the capsule upon the bacterial cell, which was actually remaining alive. Though the bacteria was heat killed, but that factor was remaining alive, and that was transforming from the S strain, heat killed S strain, to the R strain. And that was basically transforming the R strain bacteria to the smooth or the virulent strain. Okay, that means, say for example, this was the factor which was carrying the information for producing capsule. Maybe in somehow this was passing into the R strain bacteria. And as this was passing into R strain bacteria, and this was carrying information for making capsule, so it was, after passing into the R strain bacteria, it was transforming it into S strain. That means from R strain it was transforming to S strain. From the heat kill bacteria, this factor was transforming into the R strain, which was transforming the R strain to S strain or virulent strain to virulent strain. And as this was converting into virulent strain, that is why it was causing the death of the organism. And though Griffith did the whole experiment and everything, but he was not cleared about what was the chemical substance which was carrying this information and which was transforming it. That is why he gave the name transforming principle. This is very important, that why in this Griffiths experiment there is no mention about the DNA. There is no more mention about the DNA, though, but it is said like transforming principle. So Griffith said that there are some transforming principles which was basically transforming the avirulent strain to virulent strain. So these transforming principle, later on so many experiments were done by Avery, MacLeod, McCarthy, then Hershey and Chase experiments were done for the search of this transforming principle, that what was the chemical character or what was the chemical makeup of the transforming principle. But Griffith was unable to tell about the chemical makeup or chemical composition of this transforming principle. So here we are ending about the Frederick Griffiths experiment. In the next video, I am going to definitely tell you about the chemical makeup, search for the transforming principle and search for the chemical makeup of the transforming principle by different scientists in different times. So keep on watching my videos, and if you like my videos, then please don't forget to comment down below and like, share and subscribe to my channel. Thank you.